Welcome to the Bloorview Research Institute BRI Research Rounds. I'm Lauren Switzer, a research manager in the CP Discovery Lab in the Bloorview Research Institute. Before we begin, I would like to acknowledge the sacred land on which we are on today. This land is the territory of the Huron-Wendat First Nation. The Seneca is part of the Haudenosaunee Confederacy, the Mississaugas of the Credit River, and the Mississauga of Scugog Island First Nation. Today, Toronto is still the home to many Indigenous people from across Turtle Island. We recognize Indigenous land and we are committed to a path of truth and reconciliation. Reconciliation is about establishing and maintaining a mutually respectful partnership between Indigenous and non-Indigenous peoples and is a continuous process that leads to improved outcomes for Indigenous peoples. We are grateful, honored, and humbled to have this opportunity to live, work, and play in this city and this territory we call Turtle Island. Just a few housekeeping points. Please note that this webinar is being recorded and will be made available to attendees and those who registered for the event but were unable to attend. To ensure that the research talk is accessible to all audiences, we have turned on live captioning. However, you can turn off or hide the captioning if you wish by finding the closed captioning button at the bottom of your screen and clicking Hide Captions. I am now pleased to introduce our guest speaker for today's event. Dr. Darcy Fehlings is a developmental pediatrician and professor in the Department of Pediatrics at the University of Toronto. She's a senior clinician scientist in the Bloorview Research Institute at Holland Bloorview Kids Rehabilitation Hospital. She also leads the CP Discovery Lab at Holland Bloorview and has held positions such as the inaugural head of the U of T Division of Developmental Pediatrics from 2006 to 2022 and the president of the American Academy of Cerebral Palsy and Developmental Medicine in 2015. Dr. Fehlings' clinical and research work focuses on the innovation and evaluation of interventions for children with cerebral palsy, including hypertonia interventions, early detection, constraint therapy, dystonia, and pain management. She is the lead investigator of an Ontario Brain Institute integrated neuroscience discovery network focused on children with cerebral palsy called CPNet, which includes a focus on the genomics of cerebral palsy. Dr. Fehlings' talk is titled Genomic Architecture of Cerebral Palsy: How are the latest research findings impacting clinical care and creating opportunities for precision medicine? She will present her cutting-edge research using the latest technology of whole genome sequencing to investigate the genomic architecture of cerebral palsy. This work arises out of large cerebral palsy networks. She will also discuss the intersection of genomic etiological risk with clinical care, including diagnosis, genetic actionability, and enhancements in precision medicine. After her talk, we will have time for a Q&A with Dr. Fehlings, so please send your questions to the Q&A box throughout the presentation, and we'll be sure to answer your questions afterwards. Please join me in welcoming Dr. Darcy Fehlings.
Thank you so much, Lauren, and thank you everybody for joining us today for these research rounds. Please feel free to put in the chat where you're from while I get the screen up. So I'm really pleased to be here talking with you about exploring the genomic architecture of cerebral palsy, and then to interpret it in terms of the impact it's having on clinical care and opportunities for precision medicine. So here's a beautiful picture of our new renovated research institute, and going along with the research institute, we have some themes for the research institute. And this particular project that we're talking about today really feeds into the theme understanding developmental diversity. Eventually, as the research progresses forward, it also will help to shape developing new interventions for individuals with cerebral palsy. So science is always a team science, and I just wanted to acknowledge our CP Discovery Lab team here. Here's a collective group of pictures over many years. And in particular, I want to highlight Lauren Switzer, who has helped with the introduction today, but also is our research manager and really helps to ensure a high quality to all of our research projects. So thank you, Lauren, for that. So this particular slide is a copy of the slide, title slide that I presented at the American Academy for Cerebral Palsy and Developmental Medicine meeting earlier this September. And it really, again, highlights that this type of research is best done within a team science approach. So you'll see the number of authors and co-authors here that have contributed to this work. And by necessity, it really is a team. It's a team of clinician scientists who have done a lot of the background work doing the clinical phenotyping of the children with cerebral palsy, and then partnering with genomic scientists to do a deep dive into the genomic architecture of cerebral palsy. This is a long list, but I just want to highlight a few people. So Mehdi Zeri, through a postdoc work, did a lot of the deep dive into the genomic whole genome sequencing of the CP cohort. And Bank, who is also postdoc, has provided a lot of leadership around the gene burden analysis. I want to highlight Mariam Askawi, who's a pediatric neurologist from McGill University and has helped to lead the CP registry component of the work. And then Richard Wentel is co-lead of CPNet. We'll be hearing about that in a second. And in particular, highlight Stephen Shearer, who is the VP of Research at the Hospital for Sick Children and the senior genomic scientist on the project. This is, again, a team science acknowledgement of our CPNet investigator group. And many of the children that were recruited into the study came from our CPNet project, where we have clinical sites across Ontario. So this particular research project is a great example of the power of a translational neuroscience network to bridge the gap and help fuel research discoveries. So here we have basic and translational scientists who are really providing a deep dive into the genomics. We have our clinicians and clinician scientists. And we also have a stakeholder group of individuals with cerebral palsy and families. And this partnership between these three groups is very powerful, and it can help translational neuroscience networks fuel discovery for research that's meaningful to clinicians and meaningful to stakeholders. Now, I wanted to provide a little bit of background for us today. So let's talk a little bit about the definition of cerebral palsy. So cerebral palsy really is an umbrella term, and it represents a group of disorders of the development of movement and posture, causing an activity limitation. It's attributed to a non-progressive disturbance that has occurred in the developing brain, and often it's accompanied by other health comorbidities, for example, seizures, and other neurodevelopmental impacts. Now, cerebral palsy, I mentioned, is an umbrella term, a group of disorders, and within individuals with cerebral palsy, there's really a wide variation in terms of their clinical presentation. So here on this side of the slide, we see a wide variation in the physical disability, from being able to ambulate and go upstairs to requiring a walker or a wheelchair for mobility. And then on this side of the slide, I have a picture of all the different subtypes of cerebral palsy, mainly based on their neurologic examination. And you can see there's quite a bit of diversity in how the neuromotor features of cerebral palsy present. And I've got circles on ataxic and dystonic. And the reason I've circled these two subtypes is that often individuals who have a genomic contribution to their cerebral palsy and neurologic presentation are often in these two subtypes. So ataxic cerebral palsy refers to when you're having problems with balance and coordination. And dystonic cerebral palsy refers to a type of involuntary movement disorder or stiffness that we can detect on neurologic examination. So what's the history of cerebral palsy and how does this relate to our newfound information on genomics? In 1861, Professor Little, who's an orthopedic surgeon at the time, presented research to the Obstetrical Society of London, where he provided the first definition of cerebral palsy. He stated that children with the condition have an injured nervous system that results in spasticity or increased stiffness in the muscles. And he postulated that cerebral palsy was acquired at birth due to a difficult labor. So that's really an environmental postulation to the etiology of cerebral palsy. A child proceeds through a difficult labor and sustains an injury to the brain that then subsequently presents as cerebral palsy. However, when we look at our large research studies, even before we started to dive into genomics, we found that many children had cerebral palsy and they did not have this history of a difficult labor and difficult birth history. And so that really prompted clinicians and researchers to try to further understand what might be related to increasing the risk of a child developing cerebral palsy. And in the last several years, research is mounting to support the role of genetic etiologic risk of cerebral palsy. So the research objectives for this particular study was to really use the latest cutting edge technology of whole genome sequencing to annotate the full spectrum of genetic variation in individuals with cerebral palsy. Secondary objectives, we wanted to see if there were any CP phenotypic characteristics that were associated with individuals who had a genomic variation. And we were looking at variants in the gene makeup of the individual that were labeled as pathogenic or likely pathogenic in terms of linking to cerebral palsy or neurodevelopmental features of the child. And then another secondary objective was to really try to do a deep dive into the genes through a gene burden analysis to further understand how the genes being identified in individuals with cerebral palsy might be linked to a disturbance in the brain. So some of the terminology that genomics uses is a little bit complex. So I wanted to give a little bit of background on it. So we've already talked about the contributions from genetic variation to the clinical phenotypes of CP are increasingly being recognized. And the majority of studies over the last 5 to 7 years have used exome sequencing. And exome sequencing is useful in detecting SNVs or single nucleotide variants. There's also been some earlier studies that have used microarray studies, and microarray studies are good at detecting copy number variants. I'm going to tell you what those two things are in a second. So we've got our exome sequencing, we've got our microarray studies, and then if you look at the third bullet, the power of whole genome sequencing is it can detect the full range of genetic variation. It can detect SNVs, CNVs, and mitochondrial variants. And so it's powerful. It can do this all in one test. The other background to know as it relates to CP is that the rates of genomic variants associated with CP really vary depending on the genomic testing done and how the cohort was selected. So you can have a representative cohort representing all children who are coming into clinics or pulled from population-based registries, or you can form a cohort that is has a more likely chance of having a genetic variant detected. And we call that idiopathic cohorts. So the particular study that we did today was from a representative cohort. So here's a little bit more background, Genomics Background 101. So we've used trio data in this study, and that means we've collected information from the child, but also both biologic parents. And this allows us to determine if a variant is de novo or new or inherited. And if the variant is de novo, then that provides a bit more weight that the variant may be contributing to the presentation. We can extract DNA from saliva. And that's important because we're generally dealing with children, and children don't like to do blood tests. And so this is a relatively pain-free way of collecting genetic information. So I promised you definitions on SNVs and CNVs and mitochondrial variants. So I've got some pictures to go along with the definitions. So a single nucleotide variant means that there is a substitution in one letter of the base pair. And we have single nucleotide variants that occur quite frequently in our genetic makeup, but if they happen to occur in a critical area, it can impact how that gene is functioning, and that can lead to a clinical presentation. Now, copy number variants are interesting. So as I mentioned earlier, they can be picked up with microarray testing or whole genome sequencing, but not exome sequencing. And here's a picture on this side. They represent a size of a CNV is a kilobase pair or greater, so over 1,000 base pairs. So here, if we just look at our genetic makeup, just labeled in large letters A, B, C, D, E, you can have a deletion. So here we're missing B, or you could have a duplication. So here we've got 2 Bs. You could have a partial segmental duplication, or you could have an inversion. So instead of it being A, B, C, it is A, C, B. And this can impact a group of genes that may be in the area and can lead to a dosing imbalance that may have clinical significance. And now mitochondrial variants, this is something that whole genome sequencing is shining a new light on. As I mentioned, we can also look at how the mitochondrial genomic makeup is when we're doing whole genome sequencing. And mitochondria are very interesting. They are in each of our cells. And they produce, help to produce the energy in the cell. And the genetic information in mitochondrial is different than our typical genetic makeup. And here in this cell of a person, you can see that we have in blue here, just a description of the regular mitochondrial variants. But in the pink, we've represented a pathogenic variant. So one of the things that's striking here in the cell of the DNA of the mitochondria is that there's variability in their genetic makeup. The other thing that's quite interesting about mitochondria is that they're largely inherited from mother to child. And with this variability, with the child, you can have an altered ratio of variation within a cell. So in here, we're seeing that this particular cell has more pathogenic variants. And when we see this happening from mother to child, it's termed heteroplasmy. So now let's do a deeper dive into the actual research project. So here's our study design. We had 323 families representing 327 individuals with cerebral palsy, and 2/3 of the families came from CPNet, our Ontario Brain Institute funded network. And 1/3 of the families came from a national CP registry, with families coming from Montreal and Calgary. Now, the other component of the study design that I want to highlight here is that we had 2 non-CP control cohorts. One from the Canadian Healthy Infant Longitudinal Development Birth Cohort, and another from Innova. And these controls help to allow us to do a deep dive into the gene burden analysis. Now, this is the information that we have collected for the description of the individuals with cerebral palsy in our cohort. And this particular picture comes from our CPNet clinical phenotype information, but a very similar collection of information is also available through the CP registry. And we were able to pair these 2 groups together to allow for a larger study and to do a deep phenotyping dive. So within CPNet, we collect a lot of information on clinical risk factors for cerebral palsy. We do this through a health chart review, but also a parent interview. So some of the things that we're asking questions about is, is the child born early or preterm? What was the child's birth weight, and was it small or large for the age of the child? We're looking for evidence of infection or inflammation, which we know can be a risk for cerebral palsy. We also collect information on the child's neuroimaging if they've had a picture of the brain, so that we can see whether there is an area of injury or a brain malformation, or if the neuroimaging is not giving us clues. And as I mentioned, we collect saliva for a genomics deep dive, both on the child, but also both biologic parents. We collect information on their neurodevelopmental pictures. So for example, their gross motor functional status. And then for this particular study, we intersected these 3 data platforms to really allow us to generate this research. So here's our main result. In green here, we had 70% or 71% that had no clinically relevant variant that we were able to detect on whole genome sequencing. But everyone else in the color here had some findings. So in 11% of children, we identified a pathogenic or likely pathogenic variant. So if you think about that in a representative sample of children with cerebral palsy, over 1 in 10 children had a pathogenic or likely pathogenic variant. That's fairly frequent. When we do a deep dive, they're represented in the colors of blue, red, and purple. The whole genome sequencing allows us to determine the type of variant. So we had 6.4% who had single nucleotide variants. We had 3.4% who had copy number variations. And then very interesting and a unique finding to our study is the identification of 5 individuals who had mitochondrial variants that were labeled pathogenic or likely pathogenic. Now, in the yellow area, we have close to 18% who have variants of uncertain significance. And these are individuals who had some changes in their whole genome sequencing, but we did not have enough information on those variants and how they may link to cerebral palsy or other neurodevelopmental conditions. So as genomic research evolves in the future, some of these may move into the pathogenic or likely pathogenic variants. Now, our most frequent variant was COL4A. And we found that in 4 cases. I'm putting this up, it's a little bit of a busy slide, but this particular variant puts an individual at risk to have some hemorrhages or bleeding in their brain vessels. And so this is a genomic variant where it is, you're able to understand how the variant links to the brain injury, which then links to the presentation of cerebral palsy. Now, this is a busy slide, but we did a deep dive into mitochondrial variants and why they may be of interest in cerebral palsy when we were talking about what mitochondrial variants were. So if you look at the slide where I've labeled the red arrows here, and I've got these blue circles, we see in this column here, the level of the variant that's present in the mother. And you can see in these 3 cases, it's very, very low. But in this column, these are the rates that are in the child. So in each of these 3 cases, we're seeing an increase in the pathogenic variant, increasing from the mother to the child. So that heteroplasmic variability, the variant ratio is increasing in the child. And this is suggestive of a contribution of this mitochondrial variant to the clinical picture of the child. The other thing that we're able to do is, depending on the mitochondrial pathogenic variant, we can link it to what the clinical picture might be. And again, if you look here, we've labeled them and you'll see MELAS, MELAS, MELAS. And it's interesting that MELAS stands for a mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes. And so what is quite interesting in this research is that typically we have not considered mitochondrial variants within the differential diagnosis of cerebral palsy. And what this research is suggesting is that when we have an individual who has a neurologic presentation, that we should be looking at the mitochondria to see if there's a contribution from mitochondrial variants. Now, the other thing that we're able to do with this research is to look to see, are there clinical features that are recurring or give you clues that there may be a genomic variant in play? And these were the clues that came from our cohort. So we found that children with cerebral palsy who had a normal brain MRI or imaging were more likely to have a genomic variant than our individuals with CP who did not have a likely pathogenic variant. We also found a higher ratio of children who had slowness to their learning or a cognitive impairment. Similar to research that we did earlier on hemiplegic cerebral palsy, we saw a higher rate of children who were born at term compared to being born early. And then finally, children where there was consanguinity in the family history had a higher chance of having a genetic variant. So these red flags or clues can help clinicians to determine who they might test for a genetic or genomic variant. Now, this is the deep dive into the gene set burden analysis. And what we have here, just to decode the slide for you, is in red is our CP cohort. And then the green and the blue represent our comparison cohorts. And we've looked at loss of function variants, we've looked at missense variants and tandem repeats. And then we do an odds ratio to see where the gene burden is. And what's really fascinating about this particular gene burden analysis is we can see that there are a lot of important genes controlling brain development that are being identified in our individuals with cerebral palsy who have pathogenic or likely pathogenic variants. So in conclusion, for the research study, before we move to the clinical implications, we've identified pathogenic or likely pathogenic variants in at least 11.3% of a clinically representative cohort. So that means if you're a clinician, that about just over 1 in 10 children that you see in your clinic with CP will likely have a genomic or likely pathogenic variant as a contributor to their neurologic presentation. And there were multiple classes of damaging rare variants that included SNVs, CNVs, and importantly, the mitochondrial variants. So we identified mitochondrial variants in 5 cases that overlapped with the CP phenotype. The COL4A gene in our cohort was the most common, and we had 4 cases. And the other thing that I want to highlight is that in many children with CP that had pathogenic or likely pathogenic variants, we identified multiple intrinsic and environmental CP risk factors that were also identified in addition to the genomic risk. So a deep dive into the whole genome sequencing of architecture of cerebral palsy. But let's shift now to trying to understand the potential clinical impact. So there's been a lot of money spent on genomics, and the economic return has been very positive. So there's a study that's looked in the US, and it's estimated about $141 for every dollar spent. But the actual clinical application has been a little slow, and clinicians are asking, well, what does this mean when we're seeing a child with cerebral palsy? Should we order genomics testing on all children with cerebral palsy? How is it going to influence our management? So here are my perspectives on the potential areas of clinical impact for cerebral palsy. Certainly, it can impact identifying the etiologic risk and the diagnostic workup of individuals with cerebral palsy. It can inform drug therapeutics. It can enhance the understanding of the constellation of clinical findings in a child. It's certainly providing shifting perspectives on the clinical spectrum of cerebral palsy. And it can have an impact on the family as well. So let's do a deeper dive into these areas. So for the example of enhanced genomic workup, I've chosen DOPA-responsive dystonias and hereditary spastic paraplegia as examples. So let's start with hereditary spastic paraplegia. This is another group of disorders, and there's at least 70, and likely there will be more genetic subtypes of HSP that can be identified. And an early diagnosis through genomics testing would allow the clinician to integrate the knowledge about heightened weakness and presentation and course over time into their management decisions. So individuals with HSP, they present very similarly to children with spastic diplegic cerebral palsy. They have increased muscle stiffness in their legs. However, some forms of HSP can be progressive. And the other striking feature for HSP is they tend to have more clinical weakness than children with spastic diplegic cerebral palsy from other etiologies. And so being aware of the HSP diagnosis and that co-presence of weakness will potentially influence decisions. And I've given an example here around selective dorsal-rhizotomy, which is a treatment that we use to permanently reduce tone or stiffness in the legs of individuals with cerebral palsy. And when we do a selective dorsal-rhizotomy and we take away the child's extra stiffness or spasticity, it can further unmask weakness that they have. So we have to be cautious in children with HSP about proceeding with a selective dorsal-rhizotomy. It's not that we can't do a selective dorsal-rhizotomy in HSP, but we have to factor in the weakness and the knowledge of the weakness to make a good recommendation for each child. Now, dopa-responsive dystonias are another group of disorders that are commonly found in our CP cohorts. So we see HSP diagnoses in our CP cohorts. We're also finding dopa-responsive dystonias. And why this is important is that the early provision of a medicine called levodopa can reduce and in some cases eliminate the physical and cognitive impairments associated with the dopa-responsive dystonia. And the earlier you start the therapy, the better the neurologic outcome. So we know that dopa-responsive dystonias are a little bit challenging to diagnose, and we're seeing them as undiagnosed dopa-responsive dystonias in our CP cohort. So to date, it's really unknown how many hidden cases of dopa-responsive dystonia exist amongst children diagnosed with CP, but this is a really important one to catch because we have a medication that can make a big difference, and it's called levodopa. So this is just an example of a child that we were able to make a diagnosis of dopa-responsive dystonia on. This was before we were doing genetic testing. This was a clinical diagnosis where we picked up that the child had dystonia using the hypertonia assessment tool. And we queried the diagnosis of dopa-responsive dystonia. We initiated the genetic workup and genetic testing, and we started a trial of levodopa. And the gait of the child normalized within the week. So this is a really important diagnosis not to miss. So let's move on to how it can inform drug therapeutics. And I've just given you an example with the dopa-responsive dystonia. But I also wanted to give you an example of another common genetic variant that is being diagnosed in our CP cohorts called dopa-responsive CTNB1 beta-catenin deficient dystonia. And this particular diagnosis allows us to understand metabolic and neurodevelopmental pathways that may inform drug therapeutics. So the CTNNB1 gene leads to defective dopaminergic neurogenesis, which has motor and cognitive impacts. And these are some of the features that children with CTNNB1 syndrome have. So they can have an intellectual disability. They have stiffness in their legs, and this can make walking difficult. So this particular syndrome is being picked up for children who are being tested for intellectual disability, where doing a genetic and genomic workup is recommended as part of the good clinical care. In contrast, we don't have that yet for children with cerebral palsy. And because these children are routinely being tested for intellectual disability, this is why we're seeing this as a more common presentation of a genetic variant in CP. And so I talked earlier in the talk about translational neuroscience networks, and this is an example where understanding some of the basic and cellular science can really help to give you clues in terms of what you could try from a drug therapeutic perspective. So what is beta-catenin? It is an adherence junction protein that helps to mediate adhesions between the cells and anchor the actin cytoskeleton. And at the brain level, it's been shown to mediate dendritic development and is really key to the development of dopaminergic neurons. It's a beautiful stained picture of a midbrain from a rodent or marine model. And you can see here in orange, these are the green are the beta-catenin. And you can see that they're really helping to anchor the cytoskeleton. So with this knowledge that the dopaminergic neurons are impacted, we've done some clinical testing on the use of levodopa, and further research needs to be done. But in this particular case study, this child was trialed on levodopa, and we did a standardized gross motor function measure test, and there was certainly improvement in the gross motor function following the introduction of levodopa. So this is a case study, but can generate the impetus to move this forward into further research. I mentioned that genetic testing and finding can impact the family, and certainly it can have emotional impact. I've worked with many families over many years, and families are very interested in understanding how their child has developed cerebral palsy. And so having an increased understanding of the various factors that impacted their child can be helpful to the family. There's also the potential to advise and counsel around inheritance and for future children in counseling. The other impact we've seen is the impact around the diagnosis. And I want to spend a little bit of time on that aspect. So what we're seeing is that when a genetic or genomic variant is detected, some clinicians are removing the label of cerebral palsy. So it's creating a little bit of confusion around, should we use the term cerebral palsy or not? And as an international group, we have brought forward a publication that genetic or other causation should not change the clinical diagnosis of cerebral palsy. So here we're making this statement that the diagnosis of cerebral palsy is really based on clinical presentation. And if the child has a picture that fits that diagnosis of cerebral palsy, you can still have a genetic risk for cerebral palsy and use the label of cerebral palsy. And in the vast majority of children with a genetic variant, you can still keep the label of cerebral palsy. And this is important. We have many reasons to use a label of cerebral palsy. An example of one is to be able to access therapy services, for example, within the school environment. And so just a caution to clinicians that even if you identify genetic etiology, you don't need to remove the clinical diagnosis of cerebral palsy. However, I put in the bullet here for cases of progressive hereditary spastic paraplegia or DOPA-responsive dystonia or mitochondrial variants that are linked to diagnoses like MELAS or LAYS, it is likely to impact the label of cerebral palsy, and the more specific diagnosis will be utilized. So, where is the future going with genomics research in cerebral palsy? Clearly, we're still at early stages, and there's a need for large international studies. And I'm very pleased to say that there is an international cerebral palsy genomics consortium that is formed that we're part of. Richard Wentel is on their governing council, and the future will certainly include international cohort projects. The other point, though, that I want to make is that this type of research is really going to ultimately drive some new interventions, particularly as it relates to drugs. And these discoveries can be helpful. We've had very few slow development of new medication treatments for individuals with physical neurodisabilities like cerebral palsy. And so this really will help to fuel further precision medicine drug therapeutics. So to finish off, before we open it up for questions, I just wanted to leave some take-home messages for the clinicians who are listening today. So currently, we do not have a universal genomic testing program for cerebral palsy. And in the absence of a universal genomic testing, ensure you know what the MRI shows. If the MRI is normal, or if it shows brain malformations, think about doing a genetic or a genomics workup. The other thing that I wanted to encourage you to do is to not miss dystonia. So incorporate the hypertonia assessment tool into your neurologic examination. We've designed this tool to be very friendly to clinical practice. It can be integrated into your neurologic examination, and it will help you to find children who may have things like DOPA-responsive dystonias. So with that, I will stop sharing and open it up for questions and turn things over to Lauren to help facilitate the questions.
Thank you so much, Dr. Fehlings, and we are happy to answer some questions, so please feel free to send them via the Q&A box. One of the questions that has come in is, do you have any studies or projects that are planned or currently ongoing for adults with CP who might be interested in genomic testing or looking at fMRIs of those with CP?
It's a fabulous question around adults, and our particular initial work within the Ontario Brain Institute has been very focused on children. However, I'm very pleased to say that in the Ontario Brain Institute, we've received some ongoing funding, and we are going to be starting an adult cohort as part of the research objectives over the next 2 years.
Okay. To date, what impact have you seen from genetic testing for CP?
Well, that is an interesting question as well. So I am definitely seeing that clinicians are aware that genetics is needed in some children with cerebral palsy. And clinicians are wondering, should we be doing genomic testing on all children with cerebral palsy, or should we be doing it on a select group? Right now in Ontario, we don't have a universal program for genomics testing, but we're certainly seeing increased referrals or to genetics for workup. And we're seeing more children with cerebral palsy getting gene panel testing. So it's certainly impacting more testing, and we're starting to get these labels of a genetic variant associated with the diagnosis of cerebral palsy. I also have seen the impact of removing the diagnosis of cerebral palsy, and that's something that in the body of the talk, I just encourage clinicians to be cautious of taking away that diagnosis of cerebral palsy. The two can still fit together unless a more specific progressive neurologic presentation has been identified. Eventually, we're going to see more impact on actionability and findings. So again, I tried to highlight that in the body of the report, but it's an area of research that we're currently looking into is what are the actions now that we've identified a genetic variant? What are the actions that we can take?
This question relates to one of your points about the CP diagnosis. So they were wondering about your thoughts about continuing to use the CP diagnosis for some genetic causes that have a slow regressive picture like HSP. Do you think that the definition of CP should or could be revised to include these individuals?
That is a very juicy question. And I think for many reasons, clinicians, but also stakeholders are interested in doing a deep dive into the diagnosis, the definition of cerebral palsy, and how we can further clarify it. So one of the things that is creating a little bit of confusion in our working definition of cerebral palsy is the concept of it being non-progressive. And we know that as individuals age, they can have a loss of their motor function that is related to the becoming bigger and taller, but your muscle strength doesn't increase. And so their function is being impacted, even though at a brain level, there's no progression in the brain level. So some clinicians and researchers are wondering, should we take a deeper dive into that aspect of the definition? Is there a way to operationalize it so that it can encompass physical neurodisabilities that are really following a very static course in individuals when they're early in childhood? The other point I'll make is that as we do a deeper dive into genomic testing, that we're going to find the things that we have labeled as progressive neurologic disorders. I think we're going to find a broader spectrum. And so there are going to be individuals who have a milder form of that genetic variant that really very much will look like a static phenomena. And so I do think it's an important area to reflect on. And I think there's opportunity for some international consensus work around the definition of cerebral palsy to further clarify this.
All right, I think we might have time for one more question. Could genetic disorders make children more vulnerable to early brain injury? For example, is there an interplay between rare gene variants and perinatal events in the development of CP?
Yes. So when we look at our data, what's really striking, even in the children who have pathogenic or likely pathogenic variants, is that there are many environmental risk factors that are present as well. And so it appears to be an interaction between the gene and the environment. And if we look at the mitochondrial variants as an example, mitochondria are important for providing cellular energy. And so if you're having a problem with stress, then it puts the chance of running out of energy higher. So there's an example of gene environment interaction, and there's other examples. So I think one of the things that this research will help us to do is do deeper dives into what that interaction is and how it's impacting things at the brain level.
Well, I'm so sorry that we're not going to be able to answer all of the questions, but thank you so much to Dr. Fehlings for presenting your interesting research on the opportunities it has for precision medicine and clinical care. I want to thank all of you for attending today's BRI research rounds and engaging in the discussion. The BRI research rounds will be on a break for November for our 18th annual research symposium. The next BRI research rounds will be held in December with Dr. Elaine Bittis, who is a senior scientist and Bloorview Children's Hospital Foundation Chair in Pediatric Rehabilitation. Please visit www.hollandbloorview.ca/BRI research rounds to find out more details and to register. Also, if you would like to email BRI research rounds at hollandbloorview.ca to be added to the mailing list to be notified for future talks. Within the week, there will be a survey sent out shortly for your feedback on the research talk. We would really appreciate your feedback on today's event. Thank you so much again, and have a great rest of your day.